Welcome, everybody. This is our third iteration of the virtual EV Journal Club that we started about 4 years ago at Johns Hopkins. But this journal club was just held in person until a few weeks ago. So we've been really excited by the interest that's come from the international community, from all of you. And we really are humbled and grateful for your contributions to this journal club and for your interest. So after some issues that we had a week ago with some Zoom bombing, we have changed some of our security settings. So you will see that you are not able to unmute yourself right now, but you can use the chat function. So anytime you have a question, feel free to put that question right there in the chat. That should be at the bottom of your screen. There's a little chat bubble that you can use for that. And I hope that we can still have a productive discussion. I think we had a very good discussion last week, where using the chat function and then unmuting people who needed to ask the questions. So I'd like to introduce Tanina Arab, who is going to be presenting our paper this week. And so this is a very interesting paper to our lab because we're interested in the central nervous system, but I think that you can take a lot of things away from it, even if your focus is not on the central nervous system. This paper has to do with intercellular communication, and specifically in how EVs from a particular cell type can influence the function of a neighboring cell type. So without any further introduction, I'd like to turn it over to you, Tanina. Thanks so much for offering to present to us today and for facilitating this discussion. Thanks.
Thank you so much. Thank you for having me. So let's start with sharing my screen. Okay. Do you have a look on my screen?
Looks good.
Okay, so hi everyone, thank you for joining our Extracellular Vesicles Journal Club. I'm Tanina, I'm a postdoctoral fellow in Dr. Whitworth's lab, and today I'm going to discuss with you a very nice paper that has been published in the Journal of Extracellular Vesicles back on 2019, under the title Activated Human Astrocyte-derived Extracellular Vesicles Modulate Neuronal Uptake, Differentiation, and Firing. So before going ahead and having a look to the results of this paper, I would like to start with some background around astrocyte, inflammation, and central nervous system or CNS communication. So astrocytes are among cells in the central nervous system. They have been studied for several roles, including but not limited to regulation of neuronal transmitter clearance, plasticity, promoting synapse formation, and maintenance of blood-brain barrier. And being immune effector cells, they are involved in inflammation process after injury by secreting cytokines such as IL-1 beta and chemokines. These cells are also able to communicate with other cells in the CNS, including microglia cells and neurons. And this communication occurs by soluble molecules, but also by extracellular vesicles, which are secreted by almost all CNS cells, as shown here in this figure. So let's talk a little bit about these EVs. So as I mentioned before, this is a way for cell-to-cell communication. Indeed, a secreting cell is able to send functional cargo that could be protein, nucleic acid, or lipids to a recipient cell using extracellular vesicles. And depending on their size and cellular origin, there is three main groups that have been described. Apoptotic bodies, which result from cell apoptosis and are known to be biggest EVs, for which the size could reach 5 micrometers. Microvesicles, or ectosome, which result from the shedding of the plasmic membrane They are in the middle range, and their size is between 100 nanometers and 1 micrometers. And finally, exosomes, which take their origin from the luminal part of the cells after the fusion of MVBs or multivesicular bodies with the plasmic membrane. They are smaller in size, between 50 and 150 nanometers. After being studied for more than 10 years, a lot is now known about these EVs and their potential, yet some questions remain challenging. Among them, how EVs specifically target recipient cells and how EV cargo can impact the function of these recipient cells in both disease and health conditions. So today's paper addressed these questions. Let's start with why. So author designed this experiment to assess under inflammatory and non-inflammatory stimulation the effect of astrocyte-derived EVs or ADEVs on a recipient cell, which is in this study neurons. And how they did that, they use a human brain tissue from which they dissected and isolated primary astrocytes. Then these astrocytes were divided into two groups. The first group, or the control group, wasn't stimulated, and the second group undergo a stimulation under interleukin-1 beta at a concentration of 1 nanogram per ml. This stimulation lasts for 24 hours. And then prior to harvest the media, authors did an additional stimulation step with ATP 5 millimolar for 30 minutes. They explained that this step is done to conduct a complete EV secretion. These conditioned media were then used to isolate EVs or extracellular vesicles following the traditional differential centrifugation steps, following by a size exclusion chromatography. Which kind of surprised me in this protocol is the timing of the ultracentrifugation step. Indeed, they did this step for 20 hours. I'm used to this kind of timing when it's gradient ultracentrifugation, but to be honest, I never saw this timing for traditional UC when it used to be, or not to extend 3 hours. So then the first thing author did after EV isolation was to characterize them in both control and stimulation under interleukin-1 beta. And for that, they used a nanoparticle tracking analysis technology, which is one of the methodology that is using to visualize and measure nanoparticles in suspension, and this based on their Brownian motion. This kind of experiment allowed to have the particle counts and the particle sizing. They also did BCA protein assays to assess the protein concentration in their samples. So what the results show here in panel A and B is that the number of EV particles and protein concentration per ml of media is higher in interleukin-1 beta astrocyte-derived EVs compared to the control. When they report this value by protein per particle, there is no significant difference between these two groups. However, because the seeding density of astrocyte between these two groups is comparable, author concludes that that the number of EVs or particles secreted by cells was higher in interleukin-1 beta astrocyte-derived EVs compared to the control. Of note, in all the figures, it's very nicely color-coded. Blue color always refer to the control, astrocyte-derived EVs, and red color refer to the interleukin-1 beta astrocyte-derived EVs. Then they wanted to see if this stimulation have an effect on the size distribution of the isolated EVs. Again, following NTA technology, so here in panel D, they just wanted to show the traditional graphs that we obtain after NTA measurement and a capture of this particle in suspension. And in panel E and F, they reported the mean size for each group, and in F, the mode size for each group. Mode size being the majority or the population which is present in the population. What they conclude here is that using NTA, no significant difference was observed in the particle size between these two groups. The second step author did is a mass spectrometry experiment on the astrocyte-derived EVs of these both groups, and they analyzed this data using a bioinformatic tools to assess gene ontology enrichment and to have information about the cellular component, molecular function, and biological process that the detected proteins are involved to. And they also did some pathway analysis in order to have a prediction and select a candidate of interest. Let's start with the gene ontology results. So here, it surprised me a little bit, but they didn't actually study the two groups separately, but they put together all the detected proteins from the control and the interleukin-1 beta stimulated astrocyte, and it was more than 800 proteins. So what they found is that the gene ontology classification of this protein were associated with the enrichment term exosome here. So I should point out that the exosome protein classification is a historical term from when it was thought that any small EVs was an exosome. Now we know that it's not necessarily the case, so this term is somewhat misleading. And in panel B, they wanted to assess the molecular function in which this protein are enriched, and they found out that GTPase activity, MHC receptor activity, and cell adhesion molecule activity are the molecular function for which this detected protein are enriched to. And last, they did a biological process analysis, and they found that this protein are involved in cell growth, maintenance, transfer, cell communication, and immune response. And this finding kind of linked all these detected proteins to their hypothesis on EV biogenesis and target of recipient cells. What they did next is to assess the differentially expressed protein between these two groups. And for that, they did a label-free quantitative mass spectrometry. So here in panel D, they presented a volcano plot for which they raised 40 proteins in red dots that are upregulated in the interleukin-1 beta astrocyte-derived EVs comparing to the control astrocyte-derived EVs. What does it mean that is upregulated? They put the criteria of a p-value below 0.05 and fold change above 2. After that, they took these 40 upregulated proteins, and they wanted to double check with the heat map if indeed they are upregulated in the interleukin-1 beta astrocyte-derived EVs. This is what has been shown here in this heat map in panel E, that indeed these proteins are upregulated in the interleukin-1 beta astrocyte-derived EVs compared to the control. So they just double-checked and confirmed the results of the volcano plot. So then they took only the uniquely protein present in interleukin-1 beta-derived EVs group, plus the 40 proteins that were upregulated in this group, and they wanted to do some pathway analysis. This figure in the paper was kind of dense, so I chose to present the parts that are directly linked to the rest of the study. For that, I chose the panel A of figure 4, which nicely display that the proteins are involved in cell metabolism and organization, and this is due to the presence of proteins such as glucose 6-phosphate, isomerase, enolase 1, and actin. And they are also, this protein are also implicated in cellular communication, and this is mainly due to the presence of integrins proteins. And because integrins are known to control cell development, mediate cell interaction, and modulate immune signaling glial cells, author wanted to further investigate these proteins and compare the IBAC value between control and interleukin-1 astrocyte-derived EVs. So IBAC is the algorithm that author used to do the label-free quantification proteomics. And again, blue color is related to control astrocyte-derived EVs, and red color to the interleukin-1 beta astrocyte-derived EVs. They compared 10 integrin subtypes, and they presented the quantitative IBAC value comparison between these two groups. What they found is that for almost all these integrins, the quantitative proteome analysis demonstrate higher levels of these integrins in the interleukin-1 beta astrocyte-derived EVs compared to the control. So to summarize this part 2, using liquid chromatography tandem mass spectrometry, author shows that interleukin-1 beta astrocyte-derived EVs display a notable upregulation of 40 proteins compared to the control, and one protein cluster was surface proteins such as integrins, which regulate endocytic pathway. And this last finding actually derived all the step 3 of the paper. So let's have a look on this, on this last part. So in the last part of the paper, author did the in vitro studies, and they wanted to perform EV uptake experiment. And for that, they used the PKH26 lipophilic dye to stain astrocyte-derived EVs. And also, they assessed the function of this astrocyte-derived EVs on neuronal differentiation and activity. The first experiment that they did was in murine primary neurons. So after 10 days of culture, these neurons were switched in an EV-depleted media, and astrocyte-derived EVs that have been stained to PKH26 were added at a concentration of 10 micrograms. Then they tracked this uptake for 3 days. So 3 conditions were tested using the dye only, which is the control of the experiment, the stain control astrocyte-derived EVs, and the stain interleukin-1 beta astrocyte-derived EVs. I apologize for the quality of the images. We can't actually appreciate the outcome of this experiment. However, authors put some nice recorded video in the supplement, and so I chose one of them here. In the beginning of the recording, the cells are negative to the staining, and then after 3 days of tracking this uptake experiment, it turns out that some of these cells are actually taking the dye. So two quantitative measurements to evaluate the EV uptake were assessed. The first one, The first one is to measure the fluorescence intensity of cellular EVs per area among time, so day 0, day 1, day 2, and day 3. And again, it's color-coded, so in red it's the fluorescence intensity of PKH26 interleukin-1 beta astrocyte-derived EVs, and in blue line it's the uptake of the control PKH26 control astrocyte-derived EVs. And what they found is that the fluorescence intensity is higher in the interleukin-1 beta astrocyte-derived EVs compared to the control, which means that the neuron uptake these EVs preferentially comparing to the control. The second measurement that author did was the percentage of fluorescent cells, and again, data comparing to the two groups nicely shows that there is an increase of fluorescent cell percentage when they uptake PKH26 interleukin-1 beta astrocyte-derived EVs. All right. So next, they wanted to assess the implication of integrins in this process. So again, they used the primary culture cortical neurons that were switched to EV depleted media after 10 days. But this time, before adding the PKH26 astrocyte-derived EVs, they pre-treated these EVs with an integrin inhibitor, which is the peptide ARJD. And then they did a 3-day cell tracking again. So 3 conditions. The first one is the interleukin-1 beta astrocyte-derived EVs without the inhibitor, and then they wanted to test 2 different concentrations of this peptide at 0.1 micromolar and at 1 micromolar. What they found after measurement of the fluorescence intensity and the fluorescent cell percentage is that, so it's a little bit confusing this time because it's the same color, but this curve is the PKH26 AL1 beta astrocyte-derived vesicles without any peptide. And here we have this nicely superposition of the intensity measured for these 2 concentrations of the peptide. So what we can conclude here is that the pretreatment of these EVs with the inhibitor significantly prevent EV uptake by neuron under inflammatory condition because we use the interleukin-1 beta astrocyte-derived EVs. However, it turns out that the different concentration of this inhibitor doesn't have an effect on the EV uptake. So using 0.1 or 1 micromolar of the peptide doesn't change actually the result. Last, author wanted to study the impact of such an EV uptake on neuronal development by measuring the neurite outgrowth, and in neuronal activities by measuring neuronal spike firing, or said in another way, recording neuronal action potential. So again, they used murine cortical neurons, which were switched to EV depleted media after 3 days. And then they added either the control astrocyte-derived EVs or the interleukin-1 beta astrocyte-derived EVs. These neurons were incubated with, in presence with EVs for 3 days, and then fixed and labeled or stained with MP2, microtubule associated protein, and imaged using microscopy. In this experiment, authors used a dose-dependent experiment for which 1x is 1.25 microgram per ml of EVs, 2x is the double, and 4x is 4 times, so 5 microgram per ml of EVs. This is really nicely designed, and I know that in the MyZev 2018, this kind of experiment are recommended as well. So what they conclude just by these microscopy images is that, oops, no dose-dependent effects were observed in the control astrocyte-derived EVs, where in interleukin-1 beta astrocyte-derived EVs, there is an inhibition on neurite autograph in dose-dependent manner. I don't know. Actually, when I saw this pictures of microscopy, it's really hard to me to say that there is less or more, especially when we compare 2x to 1x. So these images are a different field of view. The number of cells per field of view isn't the same. So I think it's really hard to say just by showing images. However, all these experiments were processed using softwares, among them ImageG. So what data or quantitative analysis showed actually is that there is a dose-dependent reduction of neurite lates, of total surface area, and of the number of nodes in interleukin-1 beta astrocyte-derived EVs treated neurons compared to the vehicle and the controls. So they compared 1x control versus 1x interleukin-1 beta astrocyte-derived EVs, and they saw that there is a significant decrease. They found that there is a reduction in number of neurite terminals or ends, but only observed that for x of the dose that was used. However, the total number of neurite was similar among these groups. So then they assessed the activity of neurons by quantitation of number of spikes per second in panel G and the number of bursts per minute in panel K. So again, this is color-coded. Vehicle is in black, control astrocyte-derived EVs in blue, and interleukin-1 beta astrocyte-derived EVs in red. And the way they compare these neurons between them is between the same group actually. So, for example, they took an interleukin-1 astrocyte-derived EVs on day 1 that they compared to day 0, and they saw if this activity is significant or no. And then they did the same day 2 to day 0, day 3 to day 0, and so on. So what they found here is that the spike activity was significantly higher in control astrocyte-derived EVs only after 1 day, when it starts to be significantly higher only after 3 days with the interleukin-1 astrocyte-derived EVs treated neurons. However, they didn't see any significant difference either in the control astrocyte-derived EVs or interleukin-1 astrocyte-derived EVs on the neuronal burst over 5 days. So what they conclude here is that the experiment using PKH26 collectively demonstrate an enhanced intercellular transfer of EVs from reactive astrocytes, so under inflammatory condition or interleukin-1 beta stimulation to neurons, and this is significant contribution of integrins in the process. The dose-dependent experiment showed a significant inhibitory effect of interleukin-1 astrocyte-derived EVs on neurite extension and branching, but also the results indicate that the control astrocyte-derived EVs treatment accelerate neuronal maturation when there is a delay, actually, when neurons are treated with interleukin-1 beta astrocyte-derived EVs. So as a conclusion of this study, data reveal a characteristic signature in protein cargo of interleukin-1 astrocyte-derived EVs. Indeed, there is 40 proteins which were upregulated in this group. Proteomic analysis showed EV enriched enrichment of protein associated with cellular organization, cellular interaction, and inflammatory response. And furthermore, integrins seems to play a crucial role in the uptake of these EVs by neurons. And last but not least, interleukin-1 beta astrocyte-derived EVs suppressed neuric development and neuronal maturation as highlighted by the spike firing experiment. So let's discuss a little bit this results or outcome. So part one was all about NTA characterization. So unfortunately, both cell viability and starting volume information are missing in the report, which is actually crucial for experiment reproducibility across labs. I was wondering what will be the impact of ATP 5 millimolar on astrocyte-derived EV cargo. There is many reports that are using ATP actually as a stimulus, especially for microglia cells. I saw Ilaria Prada earlier joining the journal club, so I guess she will have some comment on that. And does a long ultracentrifugation step of 20 hours affect EV integrity? Unfortunately, we don't have an answer for that because we are missing some electron microscopy images. And last, how reliable is NTA technology for EV size measurement and to discriminate EVs from contaminant? So I do think that it's a good start, but cross-validation with techniques like Western blot to probe for EV markers and EV contaminants could be nicely added, as well as electron microscopy. And again, all these tips and tricks and guidelines are nicely summarized in the MyZev 2018, which was published in Journal of Extracellular Vesicles. And I think this paper is a very nice start with anyone interested in working with EVs. The second part was all about liquid chromatography tandem mass spectrometry. So again, I don't know why authors did the gene ontology studies as a pool rather than comparing these two groups separately. And this is mainly done using bioinformatic predictions. So what does really exosome, a cellular component enrichment term, really means in these databases? And again, cross-validation with bench-based experiment could be one way to go to confirm these findings. The last part in the astrocyte-derived EVs uptake. So the first question that pops up in my mind was why marine primary neurons culture when astrocytes are from human brain? So this is at the same time bad and good. It's bad because we don't know actually if the outcome will be the same with human neurons. And in the other hand, it's good because this opens doors for more cross-species application, specifically studies that use EVs as drug delivery. Is PKH26 the best EV staining or label ever? I don't know. I think some folks in our lab will have comments on that. We know that it's a lipophilic dye that stain basically all membranes. There is this aggregation of the dye. So we are open to discussion for this point. And in my point of view, there is one experiment which is missing since authors claim that under inflammatory condition, the protein on the surface of astrocyte EVs referring to integrins actually can be dynamically regulated. I think to conclude that it will be nice to do the same experiment in the presence of this integrin inhibitor, but using also control astrocyte-derived EVs and not only the interleukin-1 beta astrocyte-derived EVs. This is all for me. Thank you for your attention. I'm happy to discuss any point and to answer your question.
Well, thanks very much, Tanina, for the very clear presentation of this paper. And we've already had a very robust Q&A or comment line here in the chat function. So I'd first like to bring up a question that Phil Askenaze brought up, and then also Maria Elena had a question too about the IL-1 beta.
Mm-hmm.
So let's see here. Let me just make sure that everybody's unmuted here. So Phil, I have you unmuted if you'd like to ask your question about IL-1 beta and the relevance in the CNS.
Well, the question is why IL-1 beta was chosen. There's lots of choices. And what cell is that supposed to be coming from? I would say in new experiments, I'd want to see interferon gamma versus IL-4 or LPS as stimuli. Another aspect is to look chemically at the surface of the astrocytes to decide what activators to use.
There is a couple of nice questions here, so let me address them one by one. So why IL-1 beta? Actually, I think they're starting from what is in the literature, and they wanted just to start with one molecules. They, however, did some experiment based on ELISA assays, where they compare two different concentrations of IL-1 beta, and they measure some cytokines like CCL2 and CXCL8, and they found that under IL-1 beta stimulation, these cytokines are upregulated. So they conclude that indeed under IL-1 beta stimulation, astrocytes highlight a pro-inflammatory profile, so it's a good way to study them. Where this stimulation could come from, I think about microglia cells when I saw that. So I think in the brain, there's molecules could come from microglia cells.
On to another line of questioning that was brought up by Richa Gupta and also by Zheju Zhao, who commented on the use of the PKH26 dye. And so this is an issue that has come to the fore in recent years because all of us are using these lipophilic dyes, right, to try to track our EVs, see where they go, see what they're doing, even just be sure that they are EVs. Mm-hmm. Now the issue is that these dyes are very long-lived. So Richa, I'm going to try to unmute you so that you can ask your question, and then I'll unmute Zach as well. So Richa, why don't you go ahead and tell us what you were wanting to know?
Yes, I just wanted to know about this PKH26 dye. I assume exosomes are taken up rather quickly then, because they have been examining it for 3 days. So is it the exosomes or is just the dye that is because it's more stable in the cell and it's still there? So in the culture, are they measuring really the exosome uptake or the dye?
I'm not, I think, okay, so if I understand well your question, you're asking how, for how long the dye is stable in culture when used to stain EVs. I think this was reported in the literature. Zach, correct me if I'm mistaken, but I think it's more than 1 week, isn't it?
Yeah, dye is stable. That's what I'm saying. The dye is stable, but
yeah,
is this dye appropriate to label the exosomes and monitor its uptake?
Zach, did you have a comment on that? Because
yeah,
you mentioned, yeah.
So I was wondering, did they do so I see like 4 hours prepare a fresh batch? Do they do 3-day addition separately, or do they add one batch on time 0 and then did the fluorescent microscopy 3 times?
I do think they add one batch and then they
Okay.
follow that for 3 days.
Yeah, because once the EVs are labeled, I think they were pretty, like even with PKH, well, I haven't done 72 hours, but I think they were pretty stable. Like they will decay, but it should still have some fluorescence. And also, did they do any type of cleanup for the PKH labeling experiment?
Yeah, they did the size exclusion chromatography after staining.
Okay, gotcha. Thank you.
Yeah, so I think these are all important points because we all have to keep in mind that the stability of these dyes is really incredible. So I think Charles Lai has shown that some of the lipophilic dyes can last for a month, I mean, the third half-life. So when we have EVs in culture for 3 days, it's and what are they doing? Are they just sitting there? Are they actually transferring? Are they actually fusing with the cells? You know, I think this is a challenge. I mean, a real challenge that the field has right now. That we need to overcome with some better tools for labeling. And I know that there are a lot of people working on that, including some of the people on this call. So the interpretation of these lipid dye experiments, where we know now that that the lipid dyes can form particles themselves that resemble EVs, they can also label things that are not EVs. We need to be very, very careful with that. So, so good points brought up by everyone there. I would like to move now. There's a question that came from Erez Aitan, and that was on the possible effects of the ATP treatment, and there was some discussion of that too in the comments. So, Erez, would you like to let us know what you're thinking about ATP treatments?
Yeah, I think that ATP is also a pro-inflammatory signal that affect the neurons, and in general, it's a little bit misleading to compare the IL-1 beta to control when both of them been treated with ATP, and both of them have some pro-inflammatory effect. And I think that you saw that in some of the results, that you saw some effect of the untreated EV compared to the vehicle as well. So I know that ATP is really important to increase the yield and make the work easier, but is it justified to use it? And if so, should we discuss the fact that it's not really a control EVs, but one type of inflammation versus another type of inflammation?
Yeah, exactly. This was my concern, and actually they use ATP at a very high concentration. I saw in the literature 1 millimolar, 0.1 millimolar, when here they use 5 millimolar. So yeah, that's a little bit concerning.
Yeah, and then I think there was also a comment here in response to Erez's comment, and that was from Raha as well, who said that they noticed that the treatment was speeding up the maturation of neurons as well. Okay, so the next person that I wanted to move to was Pablo Munoz. Are you here? Trying to see if we can unmute him. There were some questions about the neurite development. I don't know if Pablo is still on the call. Yes.
Yes.
Whoops. There you go. Okay, you're unmuted. Sorry about that.
Okay, thank you. Yeah. Thank you for the presentation. It was very nice because I am very new to this topic, and I was really into the topic of the extracellular vesicle and differentiation. So I was waiting for that figure, and so I have some comments about the way that they designed that experiment. So if we are going to use or see if the extracellular vesicles induce differentiation some way, normally when we do pharmacological treatment or alteration that can affect differentiation, we do it before the axon formation, and that normally is 18 hours after plating, before 18 hours, 16 hours, and depend of the density of the neuron that you put, you can have effect of acceleration of neuron development or not. So what they, in the image, what I saw is that it wasn't too dense, the culture, which is nice, but then they treat it after 3 days. So the neurons are already past the differentiation. After 3 days, the neurons present axon, and they did only the MAP2 staining. And normally when you study differentiation, you have to do the most common staining, which is MAP2 and tau, which is staining the axon, and then we can say, does this treatment affect differentiation depending on the formation of the axon more than the neural extension? So that was the part that they put in the title differentiation, but at the end they were looking at length extension of neurite, which is made by other processes. So that was my doubt with that figure.
Thank you for your comment.
Proteomics part of the study, because when we look at the volcano plot in figure 3, so I think that the method was they had like 100,000 G-steps, then in it for 20 hours to also isolate proteins, and then they did size exclusion. And what we can see is we can see several actual populations of proteins. So when we see like this population of large increased proteins, like in positive as well as in a negative sense. And this is actually an issue I see with my own data, that the normalization is very skewed from time to time based on the proteins we co-isolate. So I think they digested something like 50 microgram of protein, and this actually leads to big problems, because like if the amount of co-isolated proteins is too large, then the amount of like exosomal proteins doesn't really change at all, just the relative amount to the total proteome. And this is very problematic with such MS studies and the actual normalization of them. So that's I'm very suspicious about that, because also like all the exosomal terms which were upregulated, like the Go terms, when we look at like any structural things, structural proteins are more or less just exosomal proteins which build a structure. So they're mostly quite stable. So seeing them as household proteins. So that's just wanted to raise my concerns about this part.
Yeah, I get your point. Actually, this is why I think NTA by itself doesn't allow you to know which size exclusion chromatography fractions you can pull together, which are EVs, which are proteins. And I think we are missing cross-validation with Western blot to probe to EV markers and to contaminant, and really know what we're pulling together and what we are not pulling together.
Bas, you had a comment too. Did you want to comment on the volcano plot?
Oh yeah, well, I totally agree with Nico, who was concerned about how the volcano plot is looking at all. You see only proteins on the right side, so it's only upregulations. That's strange to begin with. And I also agree with Nico that the Go terms, what the Go term exosomes, it was also addressed by Tanina. It doesn't really mean that much. I mean, we see all kinds of proteins in exosomes or extracellular vesicles. So all proteins, almost all proteins have the label, have the Go term exosomes in them.
Yeah.
So that makes it like a circle.
Yep. Plus here, we have to keep in mind that they put together all the proteins detected from both control and interleukin-1 beta astrocyte-derived EVs to do this Go term analysis. So
true, but I think like what you also
so that's strange to start with at all. Yeah, so it would be much more informative to look at which processes are affected when you look only at the upregulated, at the changed proteins, of course. But well, in the end, they work that out with the next analyses, and then with the integrins, which is, well, it's also a sensible step.
Yeah, great. Okay, there were a few more comments, some good comments here about some of the technical considerations in the paper, and we'll be trying to post this on YouTube soon in our channel. So thank you, Tanina, for presenting. Thank you everybody for joining, and thanks to everybody who contributed some great questions and comments and discussion. So take care everyone, and we hope to see you again next week.
Bye. Thank you.